Hello everyone and welcome to the next video. So in this video we're going to be going into detail of all of the organelles inside of the cell. So last video we talked about the cell membrane. This video we're going into the cell organelles. In the last video we're going to go into transcription, translation, and mitosis or cell division. All right. All the organelles now. So here is just the overview. Identify the cellular structures and their functions. We have our good old generic representation of the cell where we have the phospholipid membrane on the outside and then all the organelles inside. So this is a generic representation. And we're going to go through each of these organelles in this video. But what I wanted to just remind you is that in anatomy and physiology, cells, or just in life, all cells are drastically different from one another, and very few are actually these nice, beautiful round cells. They all have different numbers and different amounts of all of these organelles, so that leads us into structure and function. If you look at the structure of a cell, seeing there's a lot of mitochondria, or there's a lot of rough ER and Golgi apparatus, or a lot of smooth ER, it can give you an idea as to what the function is of that cell. So when we're talking about the organelles today, I just want to remind you that knowing the organelles are very important to understanding the purpose of the cell in the body. So a cell in the liver, lots of smooth ER. Smooth ER is an important site for detoxification and lipid production, things like cholesterol and things like that. So just a little tidbit there on different parts of the cells. So getting into the right now, the first one is kind of like a little an aside are the ribosomes. So ribosomes are the first one here. The little red dots around here. Then you see some red dots here on the rough ER. Ribosomes make proteins and via a process called translation. And we're going to go over this process in the next video. So don't worry about it too much now, but they are done at ribosomes. Some are free and some are embedded in the rough ER. And ribosomes are made of proteins and also mostly ribosomal RNA or rRNA. So we'll talk about that in the next video as well. So now we get into all the organelles that are part of what's known as the endomembrane system. This means things that are inside the cell that have a cell membrane. Anything that has a cell membrane has a phospholipid bilayer. First two are the rough and the smooth endoplasmic reticulum, or ER. So rough ER and smooth ER. Rough ER simply gets its name because it's studded with ribosomes here in red, and that makes it rough looking. So here you can see all the ribosomes in this scanning electron image of rough ER. So ribosomes make proteins. So that means proteins are made into the rough ER in here. And we're going to go over, when we talk about the Golgi, why that is. And then right with that is we have the smooth ER. Again, gets its name because there's no ribosomes. It lacks ribosomes. And like I just mentioned for the smooth ER here, this is an important thing for calcium storage. So we find a lot of this in muscle. Also detoxification. So like in the liver, lipid production, cholesterol synthesis, things like that are important for the smooth ER. And skeletal muscle, lots of calcium is stored in skeletal muscle. So we'll be talking about that a lot in that chapter too, for the release of calcium, and that leads to a muscle contracting. And then this, so here, smooth ER, kind of harder to tell what it is in an electron image right there. Next up, we have what's called the Golgi apparatus. Golgi apparatus usually goes hand in hand with the rough ER, because when proteins are made in the rough ER here, they get packaged into little vesicles and they get transported into the cis face, the inward facing side of the Golgi. Then there's these different layers, or the cisternae of the Golgi apparatus, and eventually those vesicles get to the outside, and then they can exit the cell. They can become part of the cell membrane. They can make membrane receptors here. They can make other cellular organelles like lysosomes and peroxisomes. So you can do a couple different things. But usually if you see a cell with a lot of rough ER and Golgi apparatus, that means that cell is a secretory cell, meaning it's secreting things, insulin, mucin, just to name also most things that are secreted like that. And we'll talk about examples throughout the rest of the term then too. But yeah, I like to think of it as the receiving and shipping center. So it receives the packages from the rough ER, it packages them a little better, and then so receives them, packages them, and then ships them. So it's like the UPS system of the cell. Now, the intricacies of what happens here, how it fuses and gets sent out via exocytosis, again, outside the scope of this survey course, but it is a fun, intricate process for how that works right there. Next up are the lysosomes. So lysosomes, I like to think of these as the little stomachs of a cell. So I don't have an image here for it because all they really are a phospholipid bilayer, and they have some hydrolytic enzymes inside of it. If you scroll up to the first image here, like I said, there's not much to a label right there. Same here with the peroxisomes and a vacuole then as well. Not much to them. But what do these lysosomes do? They're the stomach of the cell. So when you engulf something via phagocytosis that we talked about in the last video, those phagosomes then usually fuse with a lysosome, become a phagolysosome. And why is that? Well, there's a low pH in these, meaning it's very acidic and there's a lot of hydrogen ions. And these enzymes in there are kind of like the enzymes in your stomach where they work in that low acidic pH. And when it fuses with whatever you engulfed, it breaks it down and digests it kind of like a stomach does. And so they lyse things, they break things down. Bacteria and things like that are broken down in these lysosomes. Damaged organelles and things and all that sorts of stuff get broken down with these lysosomes so that the cells can use the parts of that for its nutrients. Next up is everyone's favorite, the mitochondria. Now, this is the plural form of saying it. A mitochondrion is a singular form. So when we all say the mitochondria is the powerhouse of the cell, to make that grammatically correct, it should be the mitochondrion is the powerhouse of the cell, or the mitochondria are the powerhouse of the cell. So there's the first grammar thing. Also, powerhouse of the cell. What does that even mean? Well, we think of it meaning ATP production. So ATP is the energy currency in the body, and that occurs via the electron transport chain, which happens to be found on these cristae folds in the mitochondria. Okay, so be it. But does every cell have mitochondria? No. Bacteria. A mitochondrion is the size of a bacteria, sometimes larger than some. So how do bacteria make energy? Do bacteria not get a powerhouse of the cell? Well, yes, it's just they have the proteins that are embedded in here in a different manner. One of the proteins is called ATP synthase. It's an enzyme that when hydrogen ions move through it, it generates ATP. That is found in bacteria. So if we were to refine the powerhouse of the cell, I would say ATP synthase, an enzyme, is the powerhouse of the cell because that's more unanimously found compared to mitochondria. So yeah, sorry to ruin that high school meme. You might know about the mitochondria there. It happens. Next up, we have the peroxisomes. Peroxisomes, perox thinks peroxide. Now, one of the byproducts of the mitochondria here in the electron transport chain is sometimes you can create oxygen-free radicals, or these reactive oxygen species are what they stand for. Those can be very dangerous to the cell. So what this peroxisome does here, again, it's a phospholipid bilayer, and then it has some enzymes inside of it. It takes those free radicals of hydrogen and converts them to hydrogen peroxide, H2O2, which then still not a great thing, but not as dangerous as free radicals here, and then eventually converts it to water via enzymes. So water, of course, isn't harmful in the correct amounts, but that's where they get their name. And there's the whole free radical theory of aging. So free radical theory of aging here, which is talking about the peroxisomes not working as efficiently, or the mitochondria producing more free radicals, leading to the breakdown of organelles, DNA damage, and so forth with age. So there is a relation between these peroxisomes and just generalized aging. They say always eat foods high in antioxidants. An antioxidant is a free radical scavenger. So kind of like peroxisomes are here. Alrighty, Last up here for the endomembrane, remember anything that has a phospholipid bilayer are the vesicles. I just included vesicles here just to remind you that there are transport vesicles moving in and out of the cell. So you have secretory vesicles here that could be secreted, they could be endocytosed, and then other vesicles that contain after being phagocytized could be a vacuole, or this contains food particles or something like that in a larger amount. A vacuole is more of a storage thing inside the cell. Alrighty, sorry for all the scrolling. Next up are the frameworks, the skeleton of the cell, also known as the cytoskeleton, conveniently enough. Now, there are 3 types of fibers we find in the cell. There are microtubules, there are microfilaments, and there are intermediate filaments. Microtubules are the big one. They're produced by these structures called centrosomes, which are, I'm not going to scroll back up to the cell image, but it's there. These provide the highways and the major framework of the cell. They can also, they're here highlighted in green in this image. So vesicles can move down the cell via, it's called vesicular transport. So when you secrete a vesicle, it needs to walk a pathway. It can help hold organelles in place, and so forth. And they're hollow, large tube with these tubulin proteins that wrap around the core. They become an important part of cell division. They are known as the spindle fibers that come out and help pull the DNA apart for cell division. And so very, very important structures here. They can also form long extensions coming out of the cell like a flagella. And then that flagella can bat. So the one mammalian cell that does that is sperm. So there are flagella. And then there are cilia. Cilia can also be these little projections. No, 1L. Cilia can also form these little projections that bat. They're smaller and they go like this and they help move fluids. Like your trachea has a ciliary escalator coming up to move mucus up and out of your lungs. Next up, we have the small. So these are the largest. Next up, we have the smallest, or the microfilaments. These are actin. Actin filaments, shown in red here, are also a structural framework of the cell, but they can also push the cell membrane and help move it. So when the macrophages and things are moving around, they can push the cell membrane out to move a certain direction, kind of like an amoeba. Also, they can create little cellular extensions called microvilli, and those are important in your digestive system for absorbing. They can increase the surface area of a cell. And then last up here, we have the intermediate filaments made of proteins like keratin. Now, they can vary in structure. So these also add another level of structural rigidity to a cell, and they can vary depending on the protein structure and so forth. But they're in they get their name because they're an intermediate size between the microtubules and the microfilaments here. So that's where they get their name, but they just add the next level structural component to the cells. Last up here, we go inside, deep into the cell where we have the nucleus. The nucleus, of course, houses the DNA. We'll talk more about DNA replication in the next video and how you actually replicate a cell and go through mitosis, but there's a phospholipid bilayer that separates the nucleus from the rest of the cell, and things get in and out of the nucleus via nuclear pores. And that's, you can see how it's connected right to the rough ER right here. Also, there's a structure labeled in here called a nucleolus. That's just where RNA is made. It's an RNA processing center. So RNA for like ribosomal RNA, there's transfer RNA. It's just where RNA is processed. Also, you see the term chromatin. Chromatin is DNA plus protein. So we have our DNA double helix, complementary base pairing, that wraps around, so DNA helix, then wraps around these proteins called histones. Think about wrapping spools on thread on a spool. They then wrap, and becomes more and more condensed. But DNA plus protein is known as chromatin, and then they condense into these little structures called chromosomes. So this side is one sequence. It's called a sister chromatid, and this side is the same exact sequence on the other side of that chromosome. So one arm is one chromosome, and 2 arms together, sister chromatids together, are 2 chromosomes. So we're going to go more into the process of the DNA, how it replicates and forms that sister chromatid. Then they get pulled apart during cell division, and that makes, in mitosis, 2 cells with genetically identical information. So you have to unwrap, you have to go through DNA replication and duplicate this side and then separate it. So we're going to go through that process and whatnot in the next video. But this is just all of the organelles in the cell. Again, very important to know because understanding the organelles helps you understand the function of a cell when we get to the various cell types in anatomy. All right, see you in the next video. If you have any questions, let me know, and bye-bye.